Hello, dear students. Today, we continue and finish a series of lectures on physics for first-year students. Also today, we continue to deal with electromagnetic radiation, or rather, we will analyze the issues related to the perception of electromagnetic radiation using the optical system of the eye. Let's analyze how optical radiation can be used using devices such as a microscope, different types of microscopes. In addition, we will also be interested in questions related to the shortcomings of the optical system of the eye and the possibility of eliminating them. And we will also look at various types of microscopes, including we will also be interested in the question of the operation of the electron microscope device. So, according to tradition, we consider the issues that we have submitted for differentiated credit. That is, 3 questions. This is the optical system of the eye. The first thing that we are going to consider today is also the disadvantages of the optical system of the eye, the resolution of the optical system of the eye. In addition, we also consider optical microscopy, image formation using a microscope, how to reduce the resolution limit, increase the resolution. Well, here's the last question. It will also be considered in our lectures today. This is an electron microscope. In other words, all these questions will be considered at the lecture today, but maybe something will remain for independent study. So, well, the first thing we need to consider today is the optical system of the eye. That is, everyone knows how the eye works. We are naturally interested in the ability of the eye to form an image on the retina. And besides, we are also interested in the refractive medium of the eye. We are interested in the question of obtaining an image of the same clear on the retina, regardless of the distance at which we consider this view. Well, in general, we can say that this is a biological device that receives light. We remember that these are electromagnetic waves. Well, the human eye is able to perceive electromagnetic waves in the visible range. Also, of course, if it were not for the lens, the retina could also fix the ultraviolet sound, but due to the media of the eye, ultraviolet sound does not affect the retina, and as if this is already the protective function of the eye, because ultraviolet would already have a negative effect on the retina. So, this is a biological device that proceeds from receiving light, its primary processing. Before we have signals, the optic nerve will flow to the eye, to the brain. Here we also show the photosensitive part of the eye, that is, the retina is removed, that is, the retina has several layers of cells. We are more interested in this from the point of view of our subject. This is the work of cones and rods. We will also talk about this in more detail later. Well, in general, we can say that the eye has a refractive apparatus or a refractive apparatus and also a light-perceiving apparatus. Sensory receptor can be called in another way. In addition, it is also possible to separately allocate a communication device. In other words, someone identifies 2 main systems of the eye, someone identifies 3. And our communication device allows us to get a clear image, regardless of the distance at which we have this object. And as for the refractive apparatus, here we consider the following refractive media. First of all, this is the cornea, where the light flux initially falls. Next comes the moisture of the front chamber. Light refraction also occurs here. The next medium we have is the lens, and the last one is the vitreous body. All of these media are centered optical system, that is, their optical centers lie on the same axis, on the optical axis of the eye. Here it is, we have the optical axis of the eye shown here. And we can consider the operation of this system as a single lens. That is, if we say that the system is a centered optical system, it is in principle possible to consider the system as a lens. That is, according to the rules, to construct an image that is obtained in this case according to the rules of the lens. In addition, there is also a visual axis, that is, it is the axis with respect to which we observe the maximum sensitivity of the eye. Then here we have the maximum perception of color, and the eye can clearly view small objects. This is the visual axis, and it passes through the optical center of the lens, and also through the yellow spot. The yellow spot we have consists mainly of brackets. And in this case, we have just observed the maximum sensitivity of the eye, the ability to view small objects. The maximum light sensation, that is, in the center of the eye, we have just the maximum perception. This is what concerns the refractive media of the eye. And also, let's now look at the refractive indices of these media. For example, here we also show the refractive indices of water and air. That is, in water we have a refractive index of 1/3300, in air 1. Well, the cornea is the first medium that refracts light in our country. It's 1/3800. The moisture of the anterior chamber has a refractive index of 1.34. The lens has a maximum refractive index of the average value of 1/41, and the vitreous body has a refractive index of 1/34. Looking at these indicators, one might think, after all, which medium we have has the maximum optical power, that is, which medium has the maximum ability to refract light. And usually, looking at the lenses, they say that yes, since the lens has the maximum refractive index, then in this case, we will also have the maximum optical power. Well, let's take a look at the next slide. We will have the maximum optical power at the cornea. We see this 43 diopters. The lens has an optical strength of 18 to 20 do, although, as we have seen, the refractive index here will be the largest. The benefit of the front camera has an optical power of 3 to 5 diopters. Well, the total optical power of the eyeball is 63 to 65 diopters, somewhere the average 60 diopters is written. In other words, we see that for some reason, our cornea is characterized by the maximum optical power. Although the refractive indices of the cornea here are shown in the figure as 1/376,000. Well, if you look at the refractive indices of the optical media of the eye, we see that they do not differ much from each other. But here the lens here in the picture generally has some small refractive index, but it doesn't matter to us right now. That is, depending on the state, of course, of the lens. In this case, we have a cornea that borders on air, and the refractive index of air is equal to 1, that is, when refracted at the medium air boundary of the cornea, light is refracted as much as possible. Because the refractive index of the cornea and air is very different for us. Further, the optical media of our eyes are characterized by almost the same ability to bend color. So their optical forces will already be less than that of the cornea, because our cornea borders on air. And the air has a much lower refraction index than that of the cornea. In other words, this explains the fact that our cornea is characterized by maximum optical power. If, of course, a person is in water, then the optical power of the cornea will already be reduced. Because the refractive index of water is 1/3300, and the optical power of the eye in that case will also be reduced. In order to get an image that is formed on the retina, sometimes a reduced eye is used. This is a model of the eye. A homogeneous spherical lens. The radius of this lens of the front surface of this lens is 6 8/10 of a millimeter. The length is 24 3 millimeters. Well, the refractive index in this case is 1 as much as 4/10. That is, this lens is used to build an image. The optical strength is about the same as the angle at 60. Well, if we use this lens, we also analyze the passage of rays inside the eye, passing through the optical media of the eye. The refractive medium, we are just about to get the image that is obtained on the retina. In this case, we have an inverted thumbnail image formed. The image will be valid. That is, the image that we get on the retina is inverted, but after processing by the brain of our nervous system. Everything falls into place for us, and we see everything as it really is. And besides, we also have here we say that an imaginary image can really be obtained using optical devices, that is, the image that is actually in the field of view, but due to the use of optical devices, an image is formed on the retina that corresponds to the object that we are considering with the help of the optical device on the previous microscope. In addition, we need to focus again on the issue of forming a clear image on the retina. If we recall the formula of the lens, here it is shown, then we can say that if we change the distance from the object to the lens, in our case it is the eye, then in order to get a clear image on the screen, that is, on the retina, we also need to change the distance from the lens to the retina. As for the human eye, this is impossible. In other words, we cannot change the distance from the lens to the retina. This is a fixed distance in some animals, for example, cephalopods. The distance between the lens and the retina changes. Some fish have this ability. That is, due to this, a clear image is obtained. As for a person, in this case, another mechanism for forming a clear image is used. It is accommodation, that is, it is a change in the curvature of the lens, a change in the optical strength of the lens. So, accommodation is the ability of the eye to form a clear image or an equally sharp image of various distant objects in the retina, and change due to changes in the optical power of the eye. Well, more precisely, the optical power of the lens changes due to changes in the curvature of the refractive surfaces of the lens. Due to this, we have a clear image formation if we consider objects at a very close distance, at a distance less than the distance of the best vision. So, what happens in the case of accommodation? That is, if, first of all, we have an object at a great distance, the eye is in a relaxed state, that is, there is no tension of accommodation, then in this case, we will have a relaxed one. The lumbar muscle, but the eye lens will be as if in a stretched state due to the tension of the ligaments. So, in this case, the ciliary muscles are relaxed, well, or the ciliary muscle can be said to be relaxed, and the ciliary ligaments will be stretched. Due to this, the lens has a flatter appearance and a radius of curvature. Its surface in this case will correspond to 10 mm and 6 mm. In the case when we consider an object at a close distance, then due to the fact that signals are received in the brain, we have reflex changes in the shape of the lenses due to the fact that the muscles will contract in this case. Well, on the contrary, the ligaments will weaken. Due to this, an image is formed on the retina, a clear image on the retina when an object approaches the eye, but at a closer distance. That is, the shape of the lens changes, it becomes more convex, and in this case, the radius of curvature of the lens surface will decrease to 5/5 of millimeters. That is, the shape of the lens will be more convex. Well, these are these distances. These are the values of the shape of the lens, its curvature of the surface. And they correspond to the situation when we have maximum accommodation observed, that is, when we bring the object as close as possible, when we can still get a clear image. That is, due to the fact that the curvature of the lens changes, we can get a clear image on the retina. The optical power of the lens changes, but the optical power of the eye also changes, that is, up to 70 diodes. And when viewing objects near the eye, well, here the same thing can be seen again, how accommodation happens. That is, if we consider at a far distance, the class is in a relaxed state. That is, the muscle of the ciliated body is weakened, but in this case, the lens is embodied in us. When we approach, on the contrary, the lens shortens. The lens becomes more convex, so we get a clear image on the retina. So, well, as for accommodation, it is also important for us to carry such a concept as the distance of the best vision. The distance at which the middle eye gives a clear image without excessive strain of accommodation. That is, when our eye is not strained, here is the minimum distance, when we do not strain our eyes and see the image up close well enough. This is 25 centimeters. Further, if we bring the object even closer, then accommodation is already required by eye strain, contraction of the ciliary muscle, and a change in the shape of the lens already in this case. In addition, we also have such a thing as a point of clear vision. In other words, this is the minimum distance at which we can still get a clear image of the object using accommodation. With age, this distance increases because the elasticity of the lens changes. That is, the closer we approach, first of all, the object, the less we have the opportunity to get a clear image. That is, the minimum point of clear vision, it is within 70 centimeters in children. And with age this distance will already increase due to the fact that the elasticity of the lens decreases with us. In addition, in order to evaluate visual acuity, we use such a concept as the angle of view. In general, there is just the concept of the minimum angle of view used to assess visual acuity. But the general concept of the angle of view is the angle formed by rays drawn from the extreme points of the object through the optical center of the eye. We have just shown here that we have an object at different distances, and we conduct rays through the extreme points of this object and the optical center, well, and we can evaluate the angle at which we see this object. The minimum angle of view, the smallest angle of view at which we will see 2 points of an object separately, 2 luminous points of an object, corresponds to 1 minute for a normal healthy eye. And just this characteristic allows us to evaluate the resolution of the eye. In this case, this angle corresponds to the distance between 2 points of the object, the luminous points of the object, which is located at a distance of 70 micrometers and is at the distance of the best vision. In this case, we will have an image of 5 micrometers in size on the retina. That is, we remember that on the retina, we will have a reduced, inverted image. Well, this distance corresponds to the average distance between the two brackets. As for the gland light-receiving apparatus, that is, the formation of a nerve impulse for processing, and the formation of visual objects. The image of visual objects that are around us, then we can say that in this case, we still have the ability to perceive electromagnetic vibrations in the length range from 760 to 380 nanometers. First of all, light is refracted by the eye and it acts on the receptors of visual cells. These are cones and rods. It causes photochemical reactions in them. That is, there is a decomposition of pigment, visual pigment. For example, if we are talking about sticks, then in this case, the brain layout decomposes. And then we form a nerve impulse that enters the brain. In this way, we form visual sensations. Well, the light-perceiving apparatus of the eye is represented by the retina. The thickness of the retina we have, if we talk about the parameters from 500 to 0.5 of millimeters. Well, as for the structure of the retina too, we can say that in our center, where we have a yellow spot, there are only cones. Then we have mostly sticks on the periphery, but the rest of the retina is filled with needles, sticks. Well, the further away from the center, the more we will observe the wand. Gauzu has about 130 million sticks, 7 million cobblers. There is also a blind spot where the optic nerves exit. There we have no photosensitive cells, that is, there are no photosensitive cells. So, well, as for the sticks, let's figure it out first. In other words, they do not provide a difference in colors, that is, the same nerve impulses are formed in us, regardless of what color a given object is characterized by. And in this case, we see objects in gray-blue color in insufficient light. That is, they are characterized by high photosensitivity sticks. They form just night vision, we can say, and are also responsible for peripheral vision. As for the cones, our cones are characterized by low photosensitivity, that is, in order for us to have the formation of a visual signal with the help of cones, the illumination must be sufficient. In other words, we do not have the opportunity, and there is no way to work with cobblers in low light, but the cobblers still have the ability to distinguish small details. That is, due to the work of the cobblers, small details were brought out, then here we will have a higher resolution. In addition, according to the simplest theory of the formation of light perception, we can say that there are three main types of cones that are responsible for red, green, and blue colors. But due to the fact that their spectral curves overlap, we can see other colors, that is, we already have a more saturated light perception. That is, due to the fact that the curves overlap, we can just get various other colors. In addition, as for the perception of objects, as we have already said, depends on the illumination of objects. Well, for example, some animals we have the ability to return light rays, that is, they have a special screen, a reflective tappet screen. That is, some have seen that animals have a glow of the eyes in cats, in dogs. This is precisely why this is explained, and due to this, there is an increase in the flow of light, which in this case form an image. A person does not have such an opportunity, but in addition, there is still an adaptation to image acquisition in low light. First of all, our pupil can change its diameter, that is, from 1/8 of a millimeter to 7.5 millimeters, that is, the pupil area increases 17 times. But if we talk about the illumination, after all, we will increase not by 17 times, but by 10 times. In addition, that is, here is the first opportunity to increase the amount of light that enters the eye. If we are in an insufficiently lit room, this is an increase in the diameter of the pupil. In addition, there is also the possibility of increasing the amount of degradable pigment in the sticks. Well, this process is quite long, that is, it can take up to about an hour, that is, maximum adaptation. That is, first there is an increase in the amount of pigment being deposited for the first 30 minutes. Then, more slowly, we adapt to a dark room, but this adaptation can last up to 50 to 60 minutes. But if we talk about the increase in the pupil diameter, then it takes only a few minutes and there is a change in the flow of light that enters the eye. But again, an increase in the size of the pupil leads to the fact that we have diffraction. And this can also change the quality of the perceived object, the quality of visual perception. Because diffraction leads to the fact that objects are distorted in us. And besides, as for photosensitivity, it decreases with age due to the fact that the optical properties of the sclera, pupil, and also perception receptors change. This concerns the photosensitive part of the eye, the ability to perceive color sensations, and the formation of a nervous linus. That is, due to the fact that light falls on the retina, the pigment decomposes in photosensitive cells, brackets, and rods. And then we already have the formation of a nerve impulse, which spreads along the optic nerve to the brain. This is what concerns the formation of the image. Well, now let's talk about the disadvantages of the optical system of the eye. Then here we have three types of disadvantages of the actual eye system shown. These are myopia, hypermyopia, metropia, and also astigmatism. Well, in the case of, for example, myopia, when there is, well, we call myopia in another way. We will have an image of an object formed in front of the retina due to the fact that our refractive media of the eye are activated by a sufficiently strong ability, so the image will be formed in front of the retina. In the case of farsightedness, on the contrary, the optical power of the eye will be less than necessary. Well, or with age, this pathology is observed, that is, the elasticity of the lens will no longer be enough for us. And due to this, the optical power of the eye will not change the curvature of the lens when viewing objects up close. Due to this, the image will be obtained for the retina. Well, astigmatism is characterized by the fact that we do not have an ideal shape of the cornea, the curvature of the lens changes. Due to this, we get a fuzzy image, that is, we have parallel rays that will not converge at one point, which in the usual case we should converge at one point. Well, in this case, the image of the point will be in the form of an ellipse, in the form of a straight line, well, depending on what astigmatism is observed in this case. Well, let's take a closer look at all these types of pathologies. So, myopia, let's look. That is, if we have a normal eye, then the image of the point will be on the retina. If you are nearsighted, that is, myopia can be both congenital and acquired. And congenital myopia is observed if our eye has an elongated shape. Then we will have an image obtained in front of the retina, and if we also have optical media of the eye have an optical force stronger than usual, then in this case we get an image in front of the retina. In order to get an image on the retina, to compensate for this ailment, we need scattering lenses. And with the help of scattering lenses, or we can say that these two concave lenses get an image for now. As for farsightedness, in this case, we have an insufficient possibility of refraction of light rays. As I have already said, with age, this pathology is observed when our lens will already be characterized by a lesser ability to change its curvature, or there may be a congenital pathology again when the shortened shape of the eyeball. This pathology can be corrected with the help of a collecting lens, or a doubly convex lens, as we see here in the figure. Then, in this case, we will have an image already formed on the retina. A person will already clearly see this image. You can use glasses, but you can also use lenses. As for the lens, here after all, we have an image formed not only in direct vision, but also peripheral vision will also correspond to the norm, that is, a clear image will be obtained. As for the glasses, only in the forward direction we get a clear image. And besides, there are also laser operations that change the curvature of the cornea, change the optical power of the eyes. Here again you can see the pathology data. In other words, in order to correct myopia, we need to reduce the optical power of the eye. Because there is excessive refraction of light rays, and in this case, the focal length of the eye increases, that is, the image gets on the retina. In the case of farsightedness, we have an image. First of all, it will initially be behind the retina. We need to increase the optical power of the eye, and in this case, collecting lenses are used. The image will then be obtained on the retina. And as for astigmatism, let's find out more. In this case, we change either the shape of the lens or the shape of the cornea and objects, as we see here. They no longer have a clear outline. That is, the light rays will not converge at one point, but in this case, they are either formed in the form of an ellipse, or a segment, or a figure of 8, depending on the degree of astigmatism. If there is a simple astigmatism, then cylindrical lenses can be used to correct it. Then in this case, a better quality image is formed. Also, again, a laser operation can be performed, or the use of lenses that allow you to fix this block already. Once again, you can also see the image formation in the case of astigmatism. So, this is what concerns. There were questions that concerned us with image formation using the visual apparatus of the eye. The next question is the use of electromagnetic radiation to obtain images of those objects that we cannot see with the naked eye. In other words, we are already talking about a microscope. First, consider a light microscope. That is, everyone knows that this is an optical device for obtaining highly magnified images of objects that we do not see with the naked eye. We are interested in the optical system of the microscope. The structure of the microscope, I think you should already know. Well, in this case, we see that the microscope contains two lenses, a lens and an eyepiece. That is, before the invention of the microscope, a lens, a magnifying glass was used, and an enlarged image was obtained with it. Well, in this case, our magnification experience depends on the focal length. And it is impossible to infinitely change the focal length, because this leads to operations, distortions, and the subject. Even if we upgrade the hand, the lens separately, it will be in this case its image will not correspond to reality. Therefore, it was proposed to use a system of two lenses, a lens and an eyepiece. That is, the object that we are considering in this case, it will be located in front of the lens beyond the focal length. Rays propagate from the object, which are equated in the lens. And then an image is formed that corresponds to the subject using a lens with a double focal length. That is, here we have initially designated objects as AB. Next, we have an ashbash image formed. This image will be closer to the eyepiece than the focal length, but in comparison with the lens, it will be located behind the double focal length. The formed image, it will be the rays in this case that correspond to it, will be refracted in the eyepiece, and further after refraction, will propagate through the optical media of the eye. Thus, on the retina, we get an image of a stroke, a 3 strokes, be 3 strokes. Given the image, it will be enlarged. Also, it will really be imaginary, and will be direct. But why is this image imaginary? Because the eye completes those rays that hit the retina before the image, that is, be 2 strokes, 2 strokes. This is the image that the eye will see. Since we don't really have a subject that would give this image, the image will be called me. That is, due to the refraction of optical light rays, in an optical device, an image is formed on our retina, which is then completed by the eye, the main brain. Before obtaining an enlarged image, we consider the object. This image will be located at the distance of the best view. Well, in this way we get an enlarged image of the objects in question. Well, here we can write down just that the lens we have creates a really inverted and enlarged image. This is the first refraction of light rays. Then there is a secondary refraction in the eyepiece, and due to this a secondary enlarged imaginary image is formed at the distance of the best vision. Well, here we have once again shown the entrance of the rays. That is, first there is refraction in the lens, a primary enlarged image is formed, then there is refraction in the eyepiece, and an already enlarged imaginary image is formed. The same thing is shown on this slide. In other words, we see that here we have a very small object, there is a refraction in the lens. Then the light rays are refracted in the eyepiece, enter the eye, and the eye completes those light rays that fall on the retina before the image of this object. The object that is being formed, the image of this object is at the distance of the best view. Magnification of a microscope, the angular magnification of a microscope, but in general. If we talk about magnification of optical devices, it is calculated as the ratio of the angle of view of an object obtained with the help of this optical device to the angle of view that corresponds to the observation of this object with the naked eye. That is, the greater the angle of view at which we see a given object with a microscope, the greater the magnification of the microscope in this case will be obtained. Since in our case, if we consider a microscope, we use both a lens and an eyepiece. Then we can say that the magnification of the microscope is determined by the product of the angular magnification of the lens and the angular magnification of the eyepiece. And if we recall the formula for these lenses, we can say that, well, for a lens, it is the ratio of the optical length of the tube to the focal length of the lens, and for an eyepiece, it is the ratio of the distance of the best vision to the focal length of the eyepiece. Then we get the following form. The optical bin of the obnose is multiplied by the distance of the best vision and divided by the focal length of the lens and the eyepiece. But again, it is possible to change the focal lengths naturally, but we cannot do this indefinitely because again there is an operation. Distortion. Therefore, a light optical microscope, it still has limitations on magnification, that is, a useful hobby of no more than about 1,000. A useful increase is also associated with the resolution of the eye. That is, we can of course get a greater fascination, but the eye will not be able to perceive this image. So, as for the next question, this is the resolution of the microscope. We have already talked about the resolution of the eyes. Here in principle the same meaning is the same. In other words, we say that this is the property of an optical system to give a separate image of two closely spaced luminous points. The resolution limit is the value of the distance between two luminous points that we see separately with a microscope. And with the help of this optical device, if we use some other device, for example, the same magnifying glass. And here we have the resolution and the resolution limit. These are inversely dependent characteristics. In other words, the greater the resolution, the lower the resolution limit. Well, in general, of course, scientists are interested in obtaining the minimum value of the resolution limit. But again, this is related if we are talking about electromagnetic radiation. This is due to the fact that we cannot get an image of objects whose dimensions are smaller than the wavelength of electromagnetic radiation. And just as for the resolution limit, we have it calculated according to the formula. This is the wavelength of optical radiation. Well, or we can talk about an electron microscope. There we can also estimate the wavelength, because we have an electron that also has wave properties. Well, if we are still talking about an optical microscope, then this wavelength is divided by a numerical aperture. And the numerical aperture in our denominator is multiplied by 2 by n. And this is an indicator for the agitation of the medium between the sample and the lens. And multiply by the alpha sign. Alpha is half of the input angle of the lens. Well, in general, the input angle of the lens is the Victor angle, that is, the angle formed by the rays, the extreme rays of the light cone entering the lens. Based on this formula, we can, of course, look at how we can reduce the resolution limit and increase the resolution. That is, we can reduce the wavelength and we can increase the numerical aperture. Well, as for the aperture angle, we can't increase it indefinitely either, because again there is an operation, a distortion. So two ways are used. An increase in resolution is a decrease in wavelength and an increase in the numerical aperture. And, as I have already said, we have a color microscope with limited abilities, that is, it has a resolution of about 200, but 250 nanometers. Maybe there are modern optics now, no more. And in this case, we already have limited possibilities, but we can again, as I said, reduce the wavelength and increase the numerical aperture. Here is the first possibility we have implemented in an immersion microscope, that is, in this case, it increases the hourly aperture due to the fact that it increases the refractive index between the lens and the object that is being considered in this case. In other words, we are filled with the space between the lens and the cover glass, a liquid that is close to glass in its optical properties. That is, it can be silane, he is a huge Stalin in oil. And in this case, then we will have more resolution, because again, if you look at the formula, our indicator in Me does not increase. That is, for air we have 1, but here it will already be more than 1. Due to this limit of the solution it will decrease. And besides, here it is shown here that we use immersion. Our light beam will not experience a complete internal reflection. As shown here, reflection does not occur objectively. And in this case, the brightness of the image increases. And as I said, the resolution of the microscope increases. This is the first way to increase the resolution of the microscope. The second is that it is possible to use a shorter wavelength. This is an ultraviolet microscope, but they are not widely used. They are used in microscopes, where we also use luminescence. We will also talk about this. Well, if we talk about the ultraviolet rays of their use, then naturally we need to take into account that our conventional optics are not transparent to ultraviolet rays. That is, we need to use special quartz glasses, quartz lenses. In addition, we also need to fix this image because we cannot perceive ultraviolet radiation. And here either photos or a theory screen are used. Well, here too, an ultraviolet radiation converter is used, the visible region of the spectrum. The first proposals to create an ultraviolet microscope were received at the beginning of the 20th century. And then on the basis of this idea that it is possible to reduce the wavelength, the development of an electron microscope took place. Well, we have an ultraviolet microscope that can work at a depth of waves from 180 to 400 per anonymous. Ultraviolet radiation, we also use a fluorescent microscope and a fluorescent microscope. It captures the glow of individual atoms and molecules, and therefore in this case the resolution will increase. Here we have an image that corresponds to the division of a cancer cell. That is, we introduce special markers. Fluorescent markers bind to molecules, and so they correspond to different long waves. We see that we have some molecules characterized by a blue glow, others are red, and the third is green. That is, there is a fixation of these lights, and then already superimposes one image on another, gets such a picture here. That is, in this case, it is possible to monitor several objects at once. Well, I have light filters to get one glow first, then another. How does a fluorescent microscope work? In other words, we have laser radiation that puts molecules in a state with an excess of energy and causes them to glow. In other words, we see that there is a laser, its radiation goes to the sample absorbs this radiation, and then fluorescence is observed. Here it passes through a translucent mirror, then passes through a light filter, which we pass only the radiation of the corresponding luminescence, and then it captures this image with the help of a detector. Here again, we need to use optics, which, if this ultraviolet radiation is exciting radiation, which only passes this radiation. In addition, also later, not so long ago, we modeled a fluorescence and an ultra-high-resolution microscope, which allowed us to obtain an image of smaller objects, that is, its resolution limits are about 20 nanometers. Well, here we have shown those objects that we could not consider before receiving this microscope. And then, after the dotted line, we have the objects that can be obtained. And here you can see exactly how the image is fixed in this case. That is, again, we use probes or biomarkers that are characterized by fluorescence. Well, here we have a fluorescent protein. We see that we can fix the glow of 2 points at a distance of 15 to 14 nanometers, that is, the resolution of the microscope of this microscope is much greater. Also works for us. That is, if we use a conventional fluorescent microscope, then we have a laser beam that causes all the cells of the object to glow. And in this case, we immediately observe the glow of all the cells that are in the excitation zone. In the case of a microscope on top of a falcon of resolution, we have a glow observed only in a narrow range, that is, about 20 nanometers there, for example. And this glow causes a laser beam again, but all the other molecules do not glow. Due to the fact that another laser beam suppresses this glow, that is, it is an extinguishing medium. And due to this we see the glow only in a very small range. The laser beam passes through the entire object, scans this object, and then the image of the entire object is already being formed. Well, here is just the description of this microscope. You can write it down for yourself. Well, with the help of this microscope, we also obtained an image or could see how nerve cells transmit signals to each other. And in this case, we can observe living objects just using an assistant professor and a high-resolution microscope. Also here we have a difference shown, that is, at the bottom, we have an image that is characterized by an ultra-high ratio microscope. At the top, a conventional fluorescent microscope. So, another opportunity to get an image of smaller objects is the use of an X-ray microscope. In other words, X-ray radiation is, we recall, electromagnetic radiation, but we will have a much shorter wavelength here. That is, one is used in an X-ray microscope from 1/100th to 1 nanometer. The resolution in theory can be achieved from to 220 nanometers, but still there are currently X-ray microscopes that have a resolution of no more than 5 nanometers. In addition, there is also the complexity of X-ray microscopy. The fact is that those media that are transparent to X-ray microscopes, they are characterized by almost the same ability to refract X-rays. Therefore, here is where the problem of the formation of an X-ray beam arose. Well, they use various mirrors, reflectors, to produce an X-ray beam. In addition, the use of a Snedgerov lens has also been proposed. These are lenses that are formed inside a material, and that's how two types of microscopes were created, a reflective X-ray microscope and a projection X-ray microscope. As for the reflective one, it is characterized by the maximum resolution of X-ray microscopes. Well, in the case of projection we have, well, here you can look at the view of such an X-ray microscope. As for the projection microscope, images are obtained here due to the fact that we have a different degree of absorption of X-rays by different atoms. And then there is illumination again, the photomulse or the photosensor is fixed. Here we have shown either a photosensor captures those rays that have passed through the object. Here we have an image of plants of a plant that was obtained using an X-ray microscope. Well, here we have a simplified scheme of the structure of an X-ray microscope. That is, what do we need to have? First, the source of the X-rays, the X-ray tube. Then we have optics, which forms an X-ray beam. Then the X-rays interact with the sample. After that, corrective optics are used, and the detector captures the image that was obtained in this case. Well, the last microscope that we need to consider, and which has the maximum resolution, is an electron microscope. So, here we have a model of an electron microscope of 1960. Of course, there are difficulties in creating images using an X-ray microscope, but the fact is that we have an electron wavelength much smaller than the wavelength of optical radiation. So in this case, we have a greater resolution and get less reduction in the resolution limit. In other words, we have the limits of resolution according to the following formula. Here, this formula includes a formula that corresponds to the wavelength of the electron. In other words, we have an electron characterized by both corpuscular and wave properties. It is precisely the wave properties that we use in this case. That is, in this case, we have a resolution limit of 0 as much as 5/10, as much as it is Planck's constant. Divide by the square root of 2. E is the charge of an electron, m is the mass of an electron, O is the voltage that forms the flow of electrons inside an electron microscope. Well, n is the refractive index of the medium. And then again, there is the sine of half of the electron angle. The first electron microscope was proposed, well, more precisely, its creation was proposed in 1931 year. And the microscope we have here is just shown this microscope in 1932 year by a Russian scientist. And this microscope was designed in 1932 year and in 1986 year. This scientist he received the Nobel Prize together with other scientists for the invention of the scanning probe microscope. He received the Nobel Prize, that is, 50 years after his invention. In addition, we also show here the input of rays in a conventional optical microscope where we use a light beam to form images. And also in an electron microscope, we use an electron as an image formation. And here, instead of conventional optical lenses, we already use electric magnetic lenses that form a flow of electrons. In addition, we also maintain a vacuum inside this microscope, because if there is ordinary air, then the interaction of electrons with vacuum will naturally be observed, and we will not get an image. In addition, an anode is used here, which accelerates electrons, and if we are talking about a transmission electron microscope, then some of the electrons will be scattered, some will interact with the object. And then we already get images either on a luminescent screen or on a photographic plate. It is characterized by a translucent electron microscope. The potential difference from 20 kilovolts to 1 megavolt can be used to accelerate electrons. Well, but the electron energy we have ranges from 80 to 200 kilos of electron volts. And if we have microscopes that use sufficiently large, high voltage, high power, then in this case, we also have the processing of sections, biological media, which are sections that we are considering in this case, either with metal salts or just metals in order to observe exactly the interaction of electrons with this considered an object. Well, and perhaps we also have, well, let's see the features and disadvantages you need to look at. That is, the need for a vacuum, I have already said. In addition, in this case, we need to use only very thin media. Sections of the drug in question are 50 tons per meter. Since we have electrons strongly absorbed in matter. Well, we also use it in order to get a contrasting image. We are impregnated with heavy metal salts. And some cellular structures can be destroyed. Due to the transmission, the work of a transmission electron microscope, some membrane organs were imaged, as well as biacus. It was possible to consider ibisons. And if we use a microscope with a resolution of less than 1 nanometer, that is, the resolution limit is somewhere 1 nanometer, it is possible to get an image of viruses also in the DNA chain. All the same is possible with the help of an electron microscope. Another disadvantage is that it is expensive to manufacture and maintain an electron microscope. In addition, it is also necessary to place an electron microscope in stable buildings, so that there is no exposure to external electromagnetic fields. Well, as I have already said, conductive coatings are also used. Gold, palladium can also be used, that is, heavy metal salami impregnation is used. If we use a low voltage, it is possible to obtain an image without processing with appropriate drugs. Well, here we have an image of a plant leaf in an electron microscope. That's it. We considered, first of all, the resolution of the eye, the optical system of the eye. We also considered issues related to the formation of images using a microscope, optical, electron microscope, and other types of microscopes. This lecture was the last physics lecture of this semester for first-year students. Thank you for your attention and goodbye.